All right, today we're here with Nikki Morgan. She is a coral scientist. She studies really interesting coral. They are different than most of the corals that we've heard about. So tell us about where these species live, Nikki, and how do you do research on them?
So the species that I work on are actually usually found at least deeper than 200 meters of depth or 600 feet thereabouts.
Wow.
And we usually do research on them with three different kinds of technology. We're either using automated underwater vehicles, which just usually take pictures of the seafloor and can be sent off for hours at a time without a lot of necessary handling by humans. There's remotely operated vehicles, which are attached to ships by fiber optic cables, and then controlled vans on the top of a ship, and then by human-occupied vehicles, aka submersibles, that at least 3 people are in usually, and go to the seafloor unattached to anything.
That is cool. All right.
I think so too.
So we have heard a lot in this class about the lifestyles of the shallow water corals with their photosynthetic symbionts, and you're talking 600 meters down, and there's not much light down there. Tell me about the lifestyle of the corals that you study.
So the corals that I study focus more on collecting items out of the water column. It's called suspension feeding or filter feeding, depending on how much they move their polyps in order to affect water motion around them. And so instead, surface phytoplankton and zooplankton would sink down and at some point hopefully get to those corals so that they can catch food particles from the water. So you can imagine that sometimes they're kind of hungry and there's not a lot of food. So they end up having very slow growth rates due to that. And they just hang out being hungry and catching whatever comes by. Okay.
And is this stuff that's raining down, is this what we call the marine snow?
Yeah, marine snow, and then marine snow is more of a bacterial and mucus component to it a little bit.
Okay.
And they can definitely eat that as well. But their preferred food is copepods that sink down.
All right, very cool. Can you tell me, are there any shallow water corals that are similar to this that we have? That like stuff we might see?
Photosynthetic corals. There's a few of them are of the Gorgonian family, like what I work with. The more soft octocorals is what they're called because they don't form those big reefs. They instead form more tree-like features. And so this one is actually photosynthetic, but one of the ones that I know a lot about from shallow water is actually of the genus Gorgonia ventellina. And it's a big, pretty purple sea fan in Florida that actually is, protected and an important habitat for a lot of animals in Florida shallow water.
Okay, I think I've seen flamingo tongues hanging out on those when I've been diving.
Yep. Preferred food of flamingo tongues.
Okay. Since you and I know what that is, can you tell everybody what a flamingo tongue is?
Flamingo tongue is a type of snail. I think it's a cone snail, and it's called as such because it's very spotted like the actual flamingo tongue, and it hangs out on the coral and just kind of eats the polyps through their little homes and leaves kind of a bare brown skeleton whenever it's done.
Okay, very cool. Now, my students are going to be hearing a lot about the threats to shallow water coral, which are famous, the bleaching of the coral, what temperature has to do with that. Do deep corals face the same threats? And if not, what are the main threats to deep corals? Are they healthy? Are they a danger?
So fishing at large scales is the major threat to deep water coral habitats, and that's both the more soft type corals that I work on, and there are some reforming corals as well that live in aphotic zones. And the fishing practices that give us a lot of the orange roughy that everyone likes, as well as Chilean sea bass, drag very heavy, large nets along the seafloor that are held open by big steel gates. And it just, it scares up whatever fish are there and also destroys anything on the bottom habitat and can also kick up a lot of sediment, which in areas that aren't being dragged can then also get choked up by sediment. So, especially in the North Pacific, in the main area where I study, international fishing in waters that are not controlled by anyone is also not controlled as to how much or they take or what they're taking. So no one really knows what kind of damage is being done, because it's just take it and leave, and it's no one's problem because it's international waters.
Okay, that sounds dreadful. I think I'm probably going to include a link to Christy Wilcox's article about that for the students. I think
Cool.
that might be eye-opening.
Good.
So tell me a little bit about oil extraction or mining, because I know there are other things that some people claim have an effect on deep water corals. Do they actually?
They can. Oil extraction is more generally an issue when there's a leak. And there's always going to be somewhat of a leak around oil extraction. So especially in areas that have really dense coral populations near the wellhead, they're most likely going to be affected with, raw petroleum products leaking out very oily, gross substances that are very damaging to the corals. And so we see a lot of that from the Deepwater Horizon blowout. A plume stayed at around 700 meters depth and traveled way across the area, and they found corals quite a few ways away that were covered in sludge. And they thankfully are recovering, but it's very slow again, and some of them did die as well in that area. So oil extraction can definitely be very, very dangerous if there is a problem, if something goes wrong. But in general, that one, at least, they tend to try to not drill into hard bottoms that the corals and sponges prefer to settle on. So because that takes a lot of effort to get down through that rock to get to everything. So they would prefer a soft bottom. So it'd be more of in the near area. If there were corals. Mining is kind of a new thing that deep sea scientists are trying to get ahead of. So at depth, rare minerals such as cobalt and manganese, and on hydrothermal vents, you've got gold and silver and copper, come out of the seawater. The pressures and the temperatures mean that they can't stay in solution like they were whenever they seeped out of the ocean crust. And these are used in a lot of our technologies today. The computers we're using to talk with, our favorite cell phones, those are necessary metals for technology. And it's cheaper to extract new metals than to recycle old technology that we throw away. So there's a very strong push to get the technology to go down to the seafloor to 1,000 meters or deeper and extract those metals from the rock. However,
1,000 meters, that means 3,000 feet or more down they're dying? Wow.
Yes. Yeah, so the machines they're building are actually pretty impressive from an engineering and technological standpoint, but they'd be better used by, instead looking at how you could use those on exoplanets by being able to survive harsh environments rather than extracting more metals from the seafloor. So the problem with the mining is that it takes a very long time for the precipitates to then form these crusts. One of the fields that they're looking for is called manganese nodules, and those grow at 0.1 millimeters per year. So a nodule about this size that would have maybe about this much cobalt that we need out of it took about 10,000 years to form. So that's not as sustainable.
Right.
Yeah. There's no way that we could then continue to harvest these nodules. Once you clear an area, it's gone.
Yeah.
And the animals grow on these nodules. So then any animal growing on that nodule is also removed.
Okay, so they're killing corals and sponges whenever they take the nodules.
Yes.
Okay.
And then so that area is then gone, and there's no recovery possible for that area, obviously, because that's just gone. And then in hydrothermal vents, they're looking at vents that are no longer active vents, so that chemosynthetic community is gone. But instead, corals and sponges have settled on that dead rock. And those, instead, they would then break off chimneys and extract the metals from that. And that causes a lot of sediment to stir up as well. So again, there's that choking out of animals that are needing to get food out of the water column. And when the sediment comes in, they usually just can't clear it very well, and they end up not getting food. Even if areas are not disturbed by mining, the larger area of effect by putting up these sediments that don't settle very quickly either, could choke out a much larger area than what's just removed due to mining.
Okay, that's dreadful. Let's talk about happier things. So when you study this, I know that you're one of the lucky people who gets to use the HOV, gets to use submersibles.
Yes. For 2 years, we were doing research collecting pieces of coral. So we don't take whole corals generally unless we find them already fallen over. And so we take pieces of the coral for genetic samples. And I'm looking at seeing how connected are populations of corals between different locations to see if an area is disturbed by mining or fishing. Is there a nearby population that could possibly allow for recruitment?
Okay.
And so I got to go down in the Pisces submersibles. There's Pisces 4 and Pisces 5. And I think I have a record of about 43 dives now with the 2 subs. Yeah, I'm in the top 10 for their scientists, but I'm not. They still have, scientists that live nearby, and we're at like 60 dives. So.
That's amazing. That's cool. So what is the deepest that you've been?
So the deepest I've been was actually not for this research. It was for my master's research on what's called Necker Ridge in the North Pacific still. And that one we went to 1,890 meters.
Wow.
Yeah.
That is cool.
So the limit is 2,000 meters for the subs. So we went just above that.
Yeah, that's awesome. Okay, so you've been deep down in the ocean. Have you ever seen any scary animals while you were down there?
So one of the scariest is honestly small squid, because then they scorch you with like mucus and whatnot and the pseudomorphs, and then so just a big cloud of stuff comes over the front of the sub, and you're like, what is it? And it's just mucus. But we have seen a lot of sharks. There's at least 5 different sharks I've ever seen, shark species that I've ever seen. But they actually are kind of more curious. Only one of them actually ran at the sub. But the most interesting that the sandpaper-like skin actually scratched the paint on the sub, and that was about it.
Okay, question.
But the only one...
Was that a blunt nose 6-gill? I'm just guessing.
No, actually, it was a Galapagos shark is what they're called.
Okay.
That one, it was definitely more energetic. The blunt nose 6 gills, I did see one of those, and it definitely just kind of goes, what are you?
Okay.
But the most aggressive animal we actually encountered was a swordfish that was hiding behind a rock and did not like us getting close enough to it, and zoomed out and swam straight at the sub. Actually got stuck in a light bar above where we look out at the views, and poor thing broke off its bill. It lost about this much of the top, and then managed to cut some hydropneumatic lines, basically, that allow us to move the arms and move lights around everything. So then once it was a slow leak, and then suddenly we lost control of cameras and arm, and it was like, oh, well, we got to go up because now we're leaking oil into the environment. That's no good. So yeah, the only time we had to abort a dive was because of a swordfish.
Okay, that's crazy. Now, obviously you don't spend all your time in the subs doing the cool stuff that everybody wants to do, that I want to do.
If I could, I would live in there for sure.
Yes. I know you do a lot of work in the lab. Can you describe what you do in the lab?
So we extract DNA from the physical samples, and that includes basically degrading them with enzymes so that the DNA itself can come out of the cells. And then I like to describe what I do as pipetting clear liquids into other clear liquids because I work with very small volumes. I don't sometimes have an idea of how much 25 microliters is, but it's in a tiny tube about this much liquid. So it's about, I work with small volumes and trying to find certain regions of DNA that allow us to look at connectivity between these populations. So a lot of PCR and what I also call magic. It's one of those things where I understand why we sometimes conflate science and magic together, because it's kind of, I put this liquid in this liquid and then stuff happens. And
That's cool. All right, so one last thing before we go. I almost forgot. Tell everybody how you make tiny cups. I know you made some for us.
It's my favorite thing to do at sea, because especially after you've been there for a couple weeks and you aren't sleeping very well because you're working like 18-hour days or anything like that. It's like an artistic moment where everybody gets their cups together and starts drawing out, like I made my own little Pisces sub. I'm not an artist. So it's really fun seeing just cups strewn everywhere that are all about, your normal size cups.
Yeah.
And so then we put them in a mesh bag outside the sub, or we can attach them to the CTD, which just does water sampling usually. And you send it down to depths, and the air in the styrofoam gets compressed. So you end up with what was a normal cup being your cute little tiny cup.
Oh, that's fabulous. Yeah, these are the ones. Yeah.
Whenever you do smaller cups, you can get really tiny little ones.
Yeah, that's fabulous. That's fabulous. And I appreciate the fact that you did that for our students. Let them decorate cups.
It was so much fun being able to do it. And a bunch of people got involved in helping me get everything organized for it. We have marine technicians that help us make sure everything's set up right.
Okay.
Whenever we send stuff down, and they were just like, they were like, making sure everything was zip tied together. Everyone loves doing shrinky cups.
That is cool. Well, in fact, it's popular enough. I used the cups you shrank for us in the Twitter hashtag Shrunken Cup Off contest.
Yeah, that actually went pretty popular.
It was. Anyway, thank you very much. I greatly appreciate what you've done for us today.
You're very welcome.
All right, take care.
Okay, thank you.
All right.
That's okay. I've been bitten. Ow, he has teeth. Ow, ow, ow. Some